Pharmaceutical history can be read as a series of escalating interventions, from plant extracts to purified molecules, from small molecules to biologics, from proteins to gene therapies. Each step has represented a more precise and more powerful engagement with the biological system's underlying disease. What synthetic biology is now proposing represents another order of magnitude change, not a drug that you take, but a living organism that you introduce, that takes up residence in your gut, reads your metabolic environment in real time, and produces therapeutic molecules on demand in response to what it detects. The concept is not pure speculation. Synlogic, a synthetic biology company, has already engineered E. coli strains designed to degrade phenylalanine in the gut of patients with phenylketonuria, a metabolic disorder in which toxic amino acid accumulation causes irreversible neurological damage if dietary restriction is not maintained from birth. The engineered bacteria detect phenylalanine concentrations in the gut environment and enzymatically convert it before it can be absorbed. The therapeutic goal is the same as lifelong dietary restriction, but the mechanism is biological rather than behavioral. The platform logic is straightforward and its extension to other conditions is technically credible. Bacteria that produce insulin-stimulating peptides in response to elevated glucose signals. Bacteria engineered to produce specific neurotransmitter precursors in patients with serotonin or dopamine deficiency states. Bacteria programmed to release bacteriocins, naturally occurring antimicrobial peptides, in response to the molecular signatures of specific pathogens. Each of these represents a therapeutic application for which the genetic engineering tools exist and the biological feasibility has been established in research settings. The paradigm shift this represents for medicine and for the patient-drug relationship is genuinely disruptive. Current pharmacology is transactional. You take a drug, it acts for a defined duration, it is cleared. The living medicine model is relational. A self-replicating biological system establishes a long-term presence in the body and continuously adapts its therapeutic output to real-time physiological conditions. That adaptive capacity is, in principle, a significant therapeutic advantage over fixed-dose pharmaceuticals for conditions that fluctuate dynamically. The risks, however, are not trivial and need to be stated with appropriate seriousness. Engineered organisms introduced into the gut microbiome do not remain neatly contained within the individual who receives them. Horizontal gene transfer, the exchange of genetic material between bacterial species, is a normal feature of microbiome ecology, and it means that synthetic genetic constructs could potentially spread to other organisms in ways that were neither intended nor anticipated. The ecological implications of releasing self-replicating engineered organisms, even into the contained environment of a human gut, are genuinely complex. Containment strategies, including engineered auxotrophy, where organisms are designed to be unable to survive outside the specific conditions of the target environment, are being developed alongside the therapeutic platforms. Whether those containment strategies are sufficiently robust for population-scale clinical deployment is a question that regulatory science and synthetic biology will need to resolve together, carefully and without the urgency that commercial timelines tend to impose.